All right, so good morning everyone. So today we reach, this is the last lectures for the cell biology. Next week you will start the molecular biology with Dr. Mahmoud. So today we're going to talk about active transport and glucose transport. So yesterday we spoke about the different types of transport. We said that we have the passive transport and we have also the active transport. And we said that the passive transport, it is the transport which does not require any energy, right? And this is a passive transport. Also, it is the movement of molecule between from high concentrations to the low concentration area. So it's down the concentration gradient. So we spoke about the three types of passive transport, which are simple diffusions, facilitated diffusions, and osmosis. All right. So today we're going to talk about active transport. So when we said active, I mean by that we need energy. All right. So we need to spend energy to move molecules. Why? Because this molecule will move against their concentration gradient from an area of low concentration to an area of high concentrations. Okay, so it's against the concentration gradient, so that's why we need to spend energy, all right, to move these molecules. All right, so to move these molecules we need proteins, all right. So these molecules, they will not move by themselves, all right? So we need assistance, we need the help of protein, membrane protein, all right, which will help these molecules to move against their concentration gradient. And these proteins, which are in the cell membrane, they have what we call ATPase activity. شو معناها ATPase activity? So usually these proteins in the cell membrane, they need to hydrolyze ATP. They need to hydrolyze the ATP to produce energy. All right. So that's why they have what we call ATPase. ATPase activity, yani hydrolysis. They have this kind of power to hydrolyze the ATP to produce energy, to allow them to transport the molecule against their concentration gradient. Okay, so ATPase activity, this is the activity that have this protein. They are capable to hydrolyze the ATP and to produce energy. Okay, واضح? Is it clear? All right, we have two types of active transport. We have the primary and you have the secondary, all right? The primary, we have to use, or the molecule will use directly the energy which is available. To move one molecule from one side to another side, it will use the ATP, for example. Okay? The secondary active transport, it is different because it will use another molecules. So it's a kind of co-transport. Okay, I'll explain. يلا كملنا. So primary active transport, I have one molecule needs to move from an area of low concentration to an area of high concentration. So it will use the energy and it will move. Okay, this is a primary. The secondary active transport, I have, for example, my molecule 1. To move from one side to another side, it needs the movement also to another molecule with it. So it's a kind of co-transport, مو لحالة. All right, so we have co-transport. For example, one molecule 1 معها ion. Okay, الion هذا شو يسوي? It will move also with the molecule, but down its concentration gradient. So the ion will move from high concentration area to a low concentration area. All right. So this movement of this ion, it will create a kind of electrical energy, all right? This electrical energy will help your molecule to move from an area of low concentration to an area of high concentration. Okay, so الموليكول هذه ما تقدر تمove only with the movement of the ion with it. Okay, لكن الion it move down the concentration gradient. However, the molecule it will move against the concentration gradient. Okay, so the movement of ion it will create this energy, this electrical energy, which will help your molecule to move. يعني there is no direct use of energy. We need the energy created by another molecule so that molecule can move. Okay? It depends. It is dependent of other thing. Okay? Clear? We will see it in the example so you can understand it. All right. All right. So we said that the molecule to move against their concentration gradient, they need help, they need assistance by protein, cell membrane protein. Okay, so these cell membrane protein, they are usually pump. Okay, they pump from one side and then they release it on the other side. Okay, they pump. All right, so these pumps راح يختلفوا in terms of what kind of molecule they will transport, all right, and they are all of them. They work by hydrolyzing the ATP. كلهم they have this ATPase activity. They are all capable to hydrolyze the ATP so that they are able to move the molecules. All right. I have 4 types: P-type, F and V-type, and ABC type pump. Okay, so P-type, for example, it is specific for sodium, potassium, calcium. F will V types, they are exclusively for the proton. With ABC types, it is mostly for toxins, drugs, big molecule. There is some for iron, but it's mostly for big molecules. Okay. So all these, we're going to study them one by one. We're going to see what are the difference between them. But all of them, they are carrier of, we can say, or they are pump, which will allow the movement of molecule against their concentration gradient. And they use the ATP, okay, to produce energy, so they will allow this movement. Okay. So let's start by the first pump, which is called P-class pump. Okay. So this P pump, ليش سميناها P pump? Because it binds a phosphate group. So a phosphate group PG, and it will attach to this pump. So that's why we call them P-class pump. Okay. And this P-class pump, of course, it will allow the movement of molecule against its concentration gradient. So the example that will make things more easier for you to understand, هي sodium potassium pump. The sodium potassium pump, it is a pump which is found in almost all the cell in the body. Okay. So how it works, this sodium potassium? The sodium potassium pump, it will allows to pump 3 ions of sodium, to pump them out of the cell, and to pump 2 potassium, to pump them inside the cell. So 3 sodium will go out, 2 potassium will go in. Okay? So this is the role of the sodium potassium pump. 3 sodium will go out of the cell, 2 potassium will go inside of the cell. Okay. So how it will work, how these 3 sodiums will go in and out. So this is your pump, your sodium potassium pump. Okay. So as you can see here, I have 3 molecules of sodium, 3 ions will come and will bind your pump. Okay. So here I am, this is your phospholipid bilayer. I am here in the cytoplasm. So this is my pump, this is my transmembrane protein. 3 sodiums, they will bind to my pump, all right? In the same times, I'm going to have hydrolysis of ATP into ADP, and I'm going to release a phosphate group. This phosphate group will come and it will bind to one of the subunits of the pump. Okay? So 3 sodium, it will bind to your pump. In the same time, ATP will be hydrolysis. What is the product of ATP hydrolysis? ADP plus P. Huh? Can 3-phosphate ولا D-phosphate plus P. So this P, which is released, it will come and it will bind to your pump. Okay? So when this phosphate, it will bind to your pump, it will change its conformation. So we have an opening from down. So we're going to have a change of conformation, and we're going to change the opening from outside, or the opposite side. Okay? And the sodium will be released outside. Okay? Clear? So once the sodium are released, 2 potassium from outside will come and it will bind your protein, your pump. Okay? These 2 potassium, once they will bind to your molecule or to your pump, the phosphate, it will be detached, it will be released. It will not be anymore attached to your protein. Okay? So once it is detached, شو راح يصير؟ نفس الشيء, I'm going to have change in the conformation. Okay? So it was open from up, so now it will be closed and it will open in the opposite side. And I'm going to release the potassium. Okay? Is it clear? Is it clear, or shall I repeat? Don't be shy or ashamed to say I didn't understand. I can repeat. I repeat. All right. It's very, very simple. All right. I have a cell membrane. I have my pump. Okay. In this. So this pump should سوي. It will pump 3 sodium from inside. It will release it outside. And it will pump 2 potassium from outside, and it will release it inside the cell. Okay? 3, 2. All right. So now how it works? I have my protein. I have my pump, my cell membrane protein. 3 sodium, they will bind to my pump. Okay? And in the same times, I'm going to have hydrolysis of ATP. I need hydrolysis of ATP because it's an active transport. Okay? So I need to use energy, right? So this energy here is ATP. So to release energy, the ATP must be hydrolyzed into ADP and phosphate. Okay? The phosphate which was released, it will come and it will bind to your pump. Okay? It will attach to your pump. So once it will attach, شو راح يصير? It changes in the conformations of your pump, and the sodium, it will be released outside. Okay? So once the sodium, it will be released, the potassium will come inside, okay, to the pump. It will bind to the sites. We're going to have the release of the phosphate. The phosphate, it will be detached from your pump. So once it will be detached, I'm going to have again a change in the conformations from the opening, from the opposite side. And I'm going to release my potassium inside the cell. Okay? Is it clear? واضح هلا? All right. So you have to know that this kind of pump, okay, it can be in the same time associated with another. It can be used as a secondary active transport. Okay, this is a primary active transport. Okay, but it can be used associated with the transport of glucose or whatever. لأنه why? لأنه we're gonna transport 3 potassium against 3 sodium, sorry, against 2 potassium, صح? So we're gonna create this electrical difference, all right, between, the extracellular and the intracellular media. So this difference, it creates energy, electrical energy. So this electrical energy can be used by another molecule to move against the concentration gradient. Okay, we will see the example later how this sodium potassium can be used to move another molecule. All right, clear? Okay. The second, I think, example, it is the hydrogen potassium ATPase. Okay, this is another pump. It is also a P class pump as well. And this kind of pump, we find it in the stomach and we find it in the kidney. Right? So you know that the stomach, we have acid inside, right? All right. So the acid, this acidity or this acidic pH that we have, it is due to this pump who pump the proton inside the stomach, inside the parietal cell, okay, inside the stomach cells, all right? So this pump, it will pump the proton inside the stomach, inside the cells. So this is which make your stomach acidic. The pH تبعها, it is acidic, all right? And this acidic pH, it allows the functioning of the enzymes which are present in your stomach, which will they help in the digestion of the food, all right? So now what happened here is that some drugs, they are, we can say, manufactured against this pump. خاصة for people who they have reflux, اللي عندهم acidity, ارتجاع ولا شو يسميوه، ولا عندهم reflux هذاك تبع عندهم acidity في المعدة. All right? So this kind of patients, we give them a proton pump blocker. So they block this proton pump in order so there is no proton goes inside مشان ننقص الأسيديتي اللي في المعدة. All right? So this is the drug, you think it's called Nexium or something like that. So people, they are taking them so مشان ينقصوا الحموضة, so the acidity in the stomach. All right? So they block this pump. Okay? This is just an example for you. So you can see how this kind of drug, they can targeting the pump which are present on the cell membrane. Okay? All right. The second types of pump, it is called F and V pump. F and V, they are exclusively for proton, no other ion, only H plus. All right? Only proton, neither calcium, potassium, no, only proton. All right? V class pump, هذه اللي تحدثنا عليها when we studied the organelle. You remember when we studied lysosome? And we said that the lysosome, the content of the lysosome, it is acidic. And we said how this lysosome, it is acidic. We said there is a pump which pump the proton from the cytoplasm وتحطه inside the lysosome. صح? This is it. This is the V class pump. Okay? So the V class pump is the pump which is responsible for pumping proton from, we can say, the cytoplasm to inside the what? The lysosome. Okay? The second type, it is the F class pump. The F class pump, it is located in the inner mitochondrial membrane. Okay? You remember when we studied the mitochondria, we said that it has an outer membrane, a space intermembrane, an inner membrane which is folded into crystia, and we have a matrix, and inside the matrix we have protons, we have enzymes, we have DNA, we have a lot of stuff, right? So this pump, it is located in the inner mitochondrial membrane, and what is its function? This function is just to pump proton from the matrix to the intermembrane space. Okay? They pump the proton من الماتريكس بتحطها في الانترممbrane space. Okay? And of course, all these pumps, they need, I repeat it again, they need ATP. They hydrolyze ATP. Always تلقاو في الصورة في ATP, it is hydrolyzed to produce energy, so they allow the movement of this ion or these protons. Okay? It's a primary active transport, so there is a direct use of ATP. Okay? Clear? No question? Do you have a question? Okay. The last types of protein pump, it is called ABC class pump, or we call it ATP cassette. So this molecule, or this pump, sorry, it has 4 domains, okay? Two transmembrane domain and 2 domains which are in the cytoplasm here, which are responsible for binding the ATP, okay? So they have AA, they are ATP binding the domain. So here the ATP will come and it will bind to your pump, okay? So this class of, we can say, of pumps, they are specific for substrates. It's like the other one. We said, for example, the P class, we have P class for sodium, we have P class for calcium. So they are specific for iron. Here is the same thing. The ATP cassette also, it is responsible also for transport of specific molecules. And as I said previously, they transport mostly big molecules like toxins, drugs, and so on. Okay, so they are really specific. They have specific substrate which are responsible for their transport. Okay. So the example which I need to tell you about it is the multi-drug resistance protein. The multi-drug resistance protein, this is an example of ATP cassette, an example of this pump. This kind of pump, it is overexpressed in the cancerous cells. Okay, the cancer cells بتصنع a lot of these pumps on their cell membrane. Okay, so الشخص البيشنت لما يروح to receive a chemotherapy treatment يعطيوه drugs. The drugs, it goes inside the cancerous cells in order to kill it, right? So what happened these cancerous cells? They produce a lot of this pump on their cell membrane. So once the drug, it will go inside, they will pump it out. بيخرجوا الدواء outside of the cells. Okay? So they will take it out. So that's why some patients, they become resistant to the chemotherapy. They're resistant, why? Because they have a lot of this pump expressed by the cell, all right, which will allows the cancerous cells to pump out the drug outside of the cell. Okay? So this is just an example to show you how these, you can say, ABC class pump, they can be used to pump drugs, all right, outside of the cell. Okay? Clear? واضح? Okay. All right. This is for the primary active transport. So primary active transport, I repeat it again, we're going to have a direct use of energy. We have direct use of ATP for the movement of the molecule against their concentration gradient. Now for the secondary active transport, as explained before or previously, secondary, it means it is dependent of the movement of an ion. So it's a co-transport, okay? So we transport 2 molecules together. So these 2 molecules, one molecule, it will move down its concentration gradient, and the second one, it will move against the concentration gradient. Okay, so أخذت الإكزامبل تبع الصوديوم. Sodium for example it will move from low concentration area to from high concentration area to low concentration area. It will create energy, what we call it electrical energy. This electrical energy will help the other molecule to move against their concentration area, يعني from low concentration area to a high concentration area. Okay? واضح? We're going to take example so you can understand better. Okay? All right. So as we said, for the secondary active transport, so we're going to transport two molecules on the same times. Okay? Not like the other one. The other one we have only one. So we're going to transport two molecules on the same time. So the transport of this molecule, it will be in the same direction. The two molecules will move to the same direction. So we call it symport. We call it symport or co-transport. If the transport of the molecules will be in the opposite direction, يعني one it goes in, the other it goes out, all right? So we call it antiport. So when I have same direction of the movement of the two molecules, I call it co-transport or symport. If I have the movement of the molecule in the opposite, one goes inside once the other one goes outside, so I call it counter transport or I call it antiport. Okay? واضح? So secondary active transport, I'm going to have movement of two molecules together. One molecule will provide the energy for the second one to move. Okay? Clear? All right. So this is, look to the picture here, or to the small video. This is an example of symporter movement, all right, which is represented by the sodium amino acid transport protein. So this kind of pump, it is a symporter, يعني we're going to move the two molecules in the same direction. So you can see here, the amino acids, it is low concentrated here in this area than inside, all right? And the sodium here, it is more concentrated in the extracellular fluid than the outside, the intracellular fluid. So what we're going to have? To have the movement, the sodium move from the high concentrations to a low concentration area, all right? This will create energy for the amino acids to be transported from low concentration area to a high concentration area. So if I put again the video, so you can see here sodium, and then I'm going to have also the release of amino acids. Okay, so both of them, they goes in the same directions. Okay, both of them, they go inside the cell. Okay, is it clear? All right. The next one, it is an example of antiport. So it's the opposite direction. One goes inside, the other goes outside. Okay. So as we can see here, I have the sodium and I have the calcium. So I have the calcium here, it is in low concentration here inside the cell compared to the sodium. So the sodium here, it is highly concentrated, so it will move from extracellular fluid to inside the cell. And the calcium, it is in low concentration, so it will use the energy of the sodium to move against the concentration gradient from low concentrated area to the high concentrated area. Okay? And the movement is opposite, so we call it antiport transport. Okay? Clear? You have a question about this secondary? واضح? All right. Okay. So we are done with talking about active transport. So keep in mind I have the primary active transport and I have also the secondary active transport. Primary, direct use of ATP energy by the pump. They have ATPase. They hydrolyze the ATP to provide them energy so they can move the molecule against the concentration gradient. With secondary active transport, I need the movement of two molecules together, so it's kind of co-transport. It can be symport, it can be antiport, all right, if they have the opposite direction, and they will use, the energy of a molecule to move the other one against its concentration gradient, okay? So this is overall what we saw so far. We need to talk also about the glucose as an example of transport, because you need it in phase 2 when you will study diabetes and so on. So the glucose, how the glucose, it is transported inside the cells. You are eating foods rich in carbs and glucose and sugary and a lot of stuff. So how this glucose, they will be stocked, we can say, in our body, all right, in order to reduce your level of glucose in the blood, all right? So the transporter of the glucose, we have two types in our body. We have GLUTs, we call the glucose transporter, and we have the sodium-dependent glucose transporter, okay? The GLUT, it's a very simple, we can say, transporter, glucose transporter, because they use what we call the facilitated diffusions. You know now what is facilitated diffusion, right? So what they will use? So they will use carrier protein to transport the glucose, inside the cell. Very simple, okay? From area of high concentration to an area of low concentration, okay? However, the second one, it is sodium-dependent glucose transported. What does it sound for you, this? It's a secondary active transport, okay? So it needs the sodium to be moved along with the glucose. So the movement of the glucose inside the cell, it is dependent of the movement of the sodium, okay? So we'll see it in the detail in the next coming slides. So let's start by the GLUTs. So the GLUTs, we have many types of this kind of glucose transporter, and these GLUTs, they differ in terms of their locations, okay? They are not localized in all the cell types. So for example, GLUT1, it is present in the brain and the red blood cells, okay? And these, they allows the uptake, so they allows, the entry of your glucose inside of these cells, okay? The second type, it is GLUT2. It is a bidirectional, we can say, the transport. شو معناها bidirectional transport? Yes, in two directions. يعني it allows the glucose to come in and also to get out of the cell, okay? So this is the other one, all is entry inside the cell, right? This one, it is different because it's bidirectional, so it will allow the in and out of your glucose. And this kind of transporter, it is expressed in the liver, pancreatic beta cells, and also in the kidney, okay? This is where we can find the GLUT2, all right? The third type, it is called GLUT3. So this GLUT3, we can say transporter, they are very sensitive to the glucose. So they can sense even, very little concentration, yani minimal concentration of glucose. They are very sensitive for it, and they will uptake this glucose inside of the cells, okay? So they can detect even very minimum, we can say, glucose which is circulating in the blood, all right? So they will uptake it. So this kind of protein or this kind of transporter, we can find them in the cells where they need a lot of glucose to work, okay? Like the neuron, like the skeletal, the muscles, all right? So all these kind of organs, they need a lot of glucose, so they have this kind of receptor to detect any glucose circulating so they can uptake it, okay? Clear? The other one is also important, which is the GLUT4. The GLUT4, it is insulin responsive. This one, it will be activated if we have insulin secreted, all right? So, عادة when you eat a lot of sugar or something, you're going to have a spike of glucose in your blood, all right? So when you have this spike, شو راح يصير? We're going to have secretion of insulin, all right? So insulin, what is its function? Is to stock your glucose inside the cell in order to reduce your glucose level in the blood. هذا شغلتها تخزين, all right? تحاول تنقص الجلوكوز which is circulating in the blood, all right? So one of the cells, they have the GLUT4. The GLUT4, it is activated by the insulin, so it is expressed in the cell membrane. So we're going to have uptake of the glucose inside of the cells. Usually these cells هما adipose tissue and the muscles. They take this glucose, all right, inside the cell, and the level of your glucose in the blood will be reduced. Okay? Is it clear? Have a question? All right. So how the glutes work? So how بيصير هالuptake? How the uptake occurs? So we said that it is facilitated diffusions. So the GLUT is kind of carrier. This is your protein carrier, all right? So I have my GLUT here, and there I have my glucose. So the glucose, it just will bind to my carrier, to the GLUTs. Glucose binds to GLUTs. The GLUTs, they will change conformation, closures from up, opening in the opposite side, and the release of the glucose. Okay, it's very simple, right? And of course, once the glucose it is released, the GLUTs return to the initial, we can say, conformations in order to collect other glucose molecule and repeat the same actions. Okay? واضح? Very simple. Is it clear? Okay. Now the second type, which is called the sodium glucose transport protein. So this, as I said, it is coupled, the transport of glucose, it is coupled to the sodium. Okay? So the glucose, it will be pumped, all right, in the same times with the sodium. Okay? It is dependent of the sodium. It is a symporter transport. معناها symporter transport? Yes, but what does it mean, symporter transport? Yes, but what does it mean? I know it's secondary active transport, but what does it? The same direction. يعني الصوديوم والجلوكوز, they will be in the same directions. Okay? So we have like 6 types of members, we can say, of the sodium glucose transporter molecules. So the نفس الشيء, they differ by their locations and what type of substrate they are uptaking. So we have the SGLT1, for example, بيكون موجود في small intestines, in the trachea and the kidney, and they allows the uptakes of glucose and galactose. SGL3 is like the GLUT3, it is glucose sensor, and so on. So you have here the examples of the members of the SGLT family. Okay. Yes. All right. So the SGLT, how they works? How they work? very simple. We said it's a secondary active transport, right? So imagine here I have, this is my intestine, okay? This is my intestinal cells. This is the extracellular. Have here my sodium, and I have here my glucose, all right? Then keep in mind, so we have the sodium, it is high concentrated outside than the inside the cell. However, the glucose, it is highly concentrated inside the cell than the outside of the cells, okay? So بما أنه it's a co-transport, so I'm going to have transport of the sodium along with the glucose. However, the sodium, it will move from an area of high concentration to an area of low concentrations, and the glucose, it will move from an area of low concentration to an area of high concentrations, okay? So they ensure the SGLT, it ensures the movement of both molecules in the same time, so it's a sym-porter transport. Like in the sodium from high concentration to low concentrations, and the glucose from low concentration to high concentrations, okay? So the sodium, it creates enough energy so that the glucose can be transported along with it, okay? واضح؟ So once inside the intestinal cell here, شو راح يصير؟ The glucose has to be distributed to the other cells. This is the glucose that you eat from the food, all right? From your diet. So it has to be distributed. So this glucose has to be released in the blood, all right? So for that, I'm going to use the other transporter, which is the GLUT, all right? So we're going to have like just a facilitated diffusions, all right, from an area of high concentration to an area of low concentration. So the GLUT will will uptake, we can say, the glucose, all right, change in the configurations or the conformations, and then we're going to have a release of the glucose in the blood, all right? The sodium, it has to be high outside than inside. So for that, I need what? How can I we can say how the sodium can exit the cell? By what? By which pump? Is it sodium and you tell me it's proton? How they will be exit? How they will exit the sodium? By which pump? Sodium potassium pump. Not proton. Proton is just H plus, not sodium. Okay? So the sodium potassium, so we're going to have a release of sodiums and uptake of 2 potassium. 3 sodium will go out, 2 potassium will go inside. Okay? Is it clear? All right, so we are done. Thank you very much. You have a questions? Yes. See you in the tutorial. This is our last tutorial, so try to make effort and come, please. Can you explain?